Hello? Hello? Okay, I think that is me live. As far as I'm aware. We are live. I'm just checking all the things I have. I mean, there's no one watching at the minute, but yes, I'm like, I want. Ooh, how does this work? Does this like just cut this live stream or? Okay, should be back. Is that it back? Yes, we're back. Okay. So, I have a biology test tomorrow. I am just as we start off, I do have some further maths to get done, but obviously the biology is coming. If you're here for the biology, it is coming. Let me just turn this a bit. So yes, I'm going to wait until I've got maybe about 4 people before starting the biology. I'm just going to kind of go through the textbook and yeah. I mean, there's currently no one watching. That's great. Hi, nobody. Is this video not like what? How does this work? Right, there's one person watching. That's nice. Oh, we're going into a bit of a. How does this work? Right, there's one person watching. Okay. There we go. 4 people are watching, so there's a bit of further math, just going to get this done as fast as possible. Well, not as fast as possible, as accurate as possible, I should say. Yeah, question 1 is done. Or question A, sorry. This is like really fun further math stuff. Oh look, there's one person watching. Hi, one person. Yeah, with angle pi over 3 radians. Okay, so that means it's at the origin. That's a nice easy one. Yeah, this shouldn't take too long. And then, we're straight, we'll be into biology by 35. And then it's past, I am guessing, let me get my angle drawn. What is it, pi over 3? You're pi over 3 here. Yes, I made this because I'm weird like this. Don't ask. That is part of the line. Pi over 3 is 60 degrees, in case you're wondering. That's the real axis, and that is the imaginary axis. There's one person watching. There's 2 people watching. Woohoo. Okay, we're almost done. I've got 1 question left of this further maths, and then we're straight into biology. But it's easy, so. Arg of z plus 1 equals 3 pi over 4. What is 3 pi over 4? It's over half, I think. Wait, let's use the calculator to let me know. 3 pi, this is times 180 divided by pi, 135 degrees. That's nice. So we are, this is a half line at -1, 0 with angle that. Okay. Yeah, there's still 2 people watching. That's nice. So let me just draw this. This is at -1, 0. let's just get that done. Oh, it's raining quite heavily. Wow, nice. Okay. Let's do this accurately. -1. I sense Amelia. Right. Our angle is 3 pi over 4. Full circle is 2 pi. Less than 180, so pi minus 3 pi over 4. My sister's outside. Quarter pi. Do I have a quarter pi marked on this? Pi over 2. What is pi over 4? This is the state of me. Pi over 4 times 180 over pi. 45 degrees, that's half of a right angle. Yes, I can do maths. I don't have a compass, that's the problem. We'll just estimate. Oh, actually, no, that's not too bad. It goes through on with a 0.01. So. Hello.
Sorry, you're not recording, are you?
Yes.
Reaction?
Okay. This half line, that's so this is just to line me up, and then it's like that. Because the half line is measured anti-clockwise. So this here is 3 pi over 4. Hope that's right. Anyway, let's go on to biology now. Feel free to comment because, you know, helps me know that people are actually engaging. If you're doing some revision, hi. If you're doing biology revision, double hi. Let's get this, Brett. Let's get this test done tomorrow. Okay, also, jetzt fangen wir an zu starten. Right, let me see. How am I gonna frame this? You've got this. Got a ruler. I don't know why I don't need this ruler. I'm using whiteboard pens. Okay, yes. Say hi if you're here. I want to know who's here. And if the people who said on Instagram that they want this are not here, I will be slightly annoyed. Because where are you? Okay. There's not enough space on this desk. I have. Let me just show you the situation here. I have a lot of books. I have some art I did. I do like that art. I will say that's some nice art. But the state of this desk. Can I push this any further off the edge? Okay, but there were 5 people watching for a split second. Hi, 5 people who were watching. Yeah, so I'm using the actual textbook, as you can see here, for this. There are 4 people. Say hi, seriously, say hi. I want to know that there are people here. Let me type that. Hello, Amelia.
Are you doing a live stream again?
Yes, Amelia. Okay, sure.
Sorry.
Say hi if you are here. Okay, Luke Gates. There's really not enough room on this desk. Okay. Water is the first bit. It's a quick one. Right, what do I know about water? Not much. Water. Okay, so you kind of need to know it has the formula H2O, which is drawn like, I'll use black for H's today. And you also need to know that the water can hydrogen bond to other water molecules, like this. You can also get this. This is how water works. Hydrogen bonds. If you want me to go into chemistry, let me know, because we've not done that in chemistry. So yes, water makes up approximately 70% of all living organisms, or 50 to 90 to be exact. Okay, so yes, that is water. That's how it works. See, it's polar, which is why it does that. It is a liquid because of the strong bonds. Hi, Joel Nixon. there's a thing about water which I saw called the latent heat. I think latent heat of evaporation. Amelia, can you since you're here, could you get me the biology revision guide? It's like in there. No, down. Yes, in between the chemistry one. Yeah.
So?
Yeah, it's like latent heat of evaporation, I think. You know this. No, not this one. No. No, I said the biology revision guide, right? You see the chemistry book? I think it's called latent heat of evaporation. It's the amount of energy.
This?
No, that's GCSE. It's a wee small one that says biology AS1. 1. Yes, thank you. Okay. Latent heat of evaporation, the amount of energy required to convert the liquid water into gas. Latent heat of evaporation. The amount of energy required to break the hydrogen bonds. That's fun fact. It's in the textbook, therefore it may come up in the exam. So yeah, I find that relatively interesting. Not really, but it exists. So I'm going to learn it. Okay.
Does Joel
Yes.
Nixon get our bus? No?
No.
Oh. Who is that then?
Bethany's brother.
No! Who gets the bus? You said it was Joel Nixon. I thought you did.
Who?
I don't know.
Who
You
gets
said
our
it
bus?
was Joel Nixon.
Did I?
Yes,
When?
you did.
When?
Like a few days ago.
Who?
On Monday. I don't know.
I don't think I ever said that. Okay. Right. Shh. Goodbye. You can leave. Well, I'm
I actually came in here for a coffee.
Well, you can't have coffee. It's 6:30.
Oh.
Okay. Goodbye.
Bye.
I'm revising for my test. And you have a test next week you could be revising for.
I have advice for it.
Have you?
Yes, I'm a good child.
Okay, do it again. Never hurts to revise more. Okay. Or you can watch this live stream on your phone.
Nah, I'm okay.
Isn't that a good idea, everyone? Comment yes if you think Amelia should leave my room and
No,
what?
I'll leave your room, but I just
I'd love you to leave my room. It'd be very useful for me to revise.
Yes, then I'll leave.
Okay, well, they're not going to say for another bit, so goodbye, you can leave for now. Right, okay, so let's actually start the stuff. Water is a solvent. This means it is capable of dissolving other things. We're trying to reconnect. Back. Yes. Are we back? And are we focused? Should be it now. Should be back. Alles gut. Sind wir gut? Sind wir gut? Yeah. Okay, yes, we're back. So yes, because water is a solvent, it is able to be the medium in which chemical reactions can occur. It's very abundant, which means chemical reactions can occur pretty easily. Okay. Yeah, so reactions take place in aqueous solution. So water, like uses kinda. We have reactions in body, particularly in cells, requires water transport. Amelia.
Nobody has said yes yet, have they?
No, because the stream crashed and because just it did. So goodbye.
Oh.
Okay, bye-bye. So yes, transport. That door is so loud. Transport of things. So I don't like just minerals, vitamins, things like that. So we've got things like glucose. It can also, it's like there is water and plasma. etc., etc. So yes, that is a good thing that water does. Okay, so it has effects, or it affects molecules based on hydrophilic and hydrophobic properties. How do I abbreviate this? Hydrophilic or hydrophobic properties. So yeah, that means it can, like, there'll be, for example, lipids, which I'll be covering today. Phospholipids will like clump together in a way so that they are not in the water because they are hydrophobic, or the tails or the heads are not. Okay, buffers and pH. So this is an easy one. There's not much about this. Buffers are things that mop up H plus or OH minus to minimize fluctuations in pH. So the main buffers are hydrogen carbonate, HCO3. Is that an ion? HCO3 minus, so there's hydrogen carbonate. We also have XPO3, which has, no, that's the compound, so it's really, like, it's just phosphate salts. They are able to like mop up. We also have amino acids. Amino acids, which we'll be doing later. Amino acid looks like this. And then whenever it is in or in pH 7, like normal body pH, what happens is that goes off there, and we get an extra one, so this becomes positively charged. Which means that this is called amphoteric. Amphoteric basically means positive side and negative side. So yeah, amino acids are amphoteric. They can make good buffers. Fun fact. Okay, next. Oh yeah, and the reason why buffers are useful is because enzymes obviously work at different things. Ions we'll come back to, because I don't know them. So now we're going to start organic molecules. Okay, so our first molecule is, got 2 people watching. It'd be very nice if any of you said hi, because I'm feeling a bit lonely here, you know, me by myself with my phone. Can I turn my torch on to lighten it up a little bit? No, it cannot. What happened to all the people who decided to say yes? Right, this is not working. Camera enhance. Let me see. I might be able to lighten it up a bit. I don't know how these things work. It's a bit like strange white balance. Shade. Twilight. What does this look like? Is it changing whatsoever? Warm. Oh no. Exposure. Is that working? Is this slightly brighter? Oh, okay. That's a lot brighter. And we're not focused. There we go. Okay. Alright, let's start back. So, it's now slightly brighter, which is nice. Molecules. The first one is carbohydrates. And carbohydrates are split into 3 sections, or 3 main types. We have mono, di, and poly, and they are all, like the general term, saccharides. So this is like a diagram in front of me, but I'm trying to do it by memory. Oh no, it is. So there's an H here, and there's an OH here, and it's just the H here. H, OH, ho, H, and then this is the thing that makes them different. So alpha glucose is this. Alpha glucose, as you can see, both sides are the same. Both sides are identical. Beta glucose, there's no one watching, apparently, according to YouTube. No data? What? This is happening again. Come Sean. Can I change my latency? How do I change my latency to make it stop? Stream health. Stream, hello? Oh, okay, we're back. That's fine. Okay, yes. So this is alpha glucose. I remember it in this way. So like, kind of thing. And then you're gonna have bo. So it's like basically and then beta can't be so and then bo. So then beta glucose looks like this. As you can see, like beta glucose alternates in its things. Alpha glucose just has the one that's flipped like that. So it's alpha and beta glucose. These can combine together to form many different things, but that did not stick to the table that time. And let's get rid of all that. Okay, but glucose is not. Oh, that's tragic. Glucose is not the only monosaccharide. We also have fructose, and it looks a bit like this. If I can remember, fructose is a hard one to draw. It's pentagonal. I have it drawn somewhere, don't I? Fructose is like, and then see that, and then we also have H. Oh, I think that's fructose. Am I right? Oh, I was very close. So that's fructose. That there is literally just CH2OH in reverse. Yes, that is fructose. Can I delete this comment because remove? Go. Okay. That's better. Yeah, so this is a fructose molecule. It's also a it may look like it's pentose, but it's actually hexose because it has 6 carbons. So that's just kind of the names that are given to them. Okay. Those are the only ones you really need to know how to draw. So then we go into disaccharides. That's the next part. It's whenever 2 monosaccharides come together. So we have, let's draw 2 glucoses side by side. So we have CH2OH. O. Sometimes I'll draw like this. Give it nice wide things here, and then we have AO. Then this side, it is the same, except you call it HO because opposite, and then H, O, H, 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 O, H. Okay, here we have glucose. I'm going to draw another one here. But this I'm going to call it a HO. Just because single bond C, O, single bond C, single bond C. Then we have HOH. We have out here, we have CH2OH, HOH. This is all quite repetitive. So here we have two glucoses side by side. Then what happens is the glucoses are like, oh, this is cool. Let me just, they don't like, let's change our colors. Oh look, let's bring these two things together. Okay, and then I'm gonna rub it out. Let's give out an H2O, and the oxygen that remains goes in the middle, and we have a glycosidic bond. So this here is called glycosidic bond, and it is formed by a condensation reaction. So condensation reaction, water is given out, like whenever condensation on the windows, it's not actually condensation, it's just liquid, it's just water vapor that like liquidizes, cools down, so it liquidizes. But condensation is the process of gaseous water turning into liquid water, but this is a condensation reaction, that's what it's called. The water that's stored in there comes out like this. The opposite is also a possibility, where we have something going in, we have our H2O going in. The glycosidic bond is broken by this reaction. So we get this. The O is back here, with the H goes back, and we have our HO there, and they're no longer separate. That's what happens when we add your H2O back in, and instead of a condensation reaction, that is now called a hydrolysis. Hydrolysis reaction, breaking apart by hydro is water. So that is that. And obviously different monosaccharides can come together to form different disaccharides. The main ones you need to know are, so that thing I just drew there was maltose. So we have alpha glucose plus alpha glucose equals maltose. We have alpha glucose plus fructose equals sucrose. I'm gonna restart that because I always forget and think sucrose is the word by itself. And then we have galactose plus glucose. I can't remember which one it is. I'm just going to say glucose. Do you think you need to know exactly what it is? Makes lactose. And then you could be asked, where do these sugars come from? Or like, what are they used in? So lactose is kind of an easy one. Milk sugar. Pretty much it's the sugar that is inside milk. The sugar that like people can be intolerant to, lactose intolerance. Fun fact, oh, that was a bit violent. Lactose intolerance is 80% of the world. 20% of the world are mainly found. So we have like America here. Amazing drawing, I know. And then South America there. Oops. And then we have like, here's Europe and Asia. Here we are. Look at me being a good drawer. Lactose intolerance is primarily found here and here and also here. Those are the places with the highest amount of non-lactose intolerance. So basically the whole world is pretty much lactose intolerant. Our bodies were not made to be able to digest milk. However, the faulty gene is carried in a lot of people in the Northern Hemisphere, primarily UK. Sucrose is table sugar. Sugar we all know and love. and maltose sugar is malt sugar. I had a usage for it. What was my usage? What was my usage for thing? It'll be in this book. Oh, I shook you again. You must be so shooketh. Okay. You get to see my extremely highlighted book. Where is the thing that I'm looking for? Might not be in here. You can, because I don't know if you have to know what they're all for. Google is our friend and sometimes our only friend. I think malt sugar, that's what it is, but what is maltose? Google will tell us. Maltose, sugar breakdown of starch. Yeah, what is maltose made of? It's made from 2 glucose units. Maltose disaccharide. Yeah. Maltose. Where are we getting it? Malt sugar. Yes. Okay, yes. Malt sugar. It's basically sugar from malt. Malt is like a plant thing. Sugar from malt. I think it's using brewing. That's the usage of that one. Yes, maltose is formed whenever starch is digested. Starch is broken down into maltose, pretty much. Okay, so those are our... Oh wait, there's one other thing about disaccharides. The bond, well, there's two actually. The bond is named after whereabouts. So if we draw our... This is what you call the basic... Well, just like the... It's pretty much the most basic ring structure that you can get. Yeah, so that is pretty much what starch looks like, but you can do that as well. The bond formed here is called, so the bond formed between these, we'll draw it like this, is so we count carbons here, 1, 2, 3, 4, and this is carbon 1, so it's called a, and these are both alphas by the way, it's called alpha 1, 4 glycosidic bond. That is important. Alpha, you can have a dot in there. Alpha 1 for glycosidic bond. That's what it's called because it's attached to the 4th carbon and the 1st carbon. Then obviously beta 1, 4, the ones that happen up there are 6 ones. And also the equation for forming a disaccharide. That's going to really annoy me. We have so C6H12O6 plus another C6H12O6 goes to. So remember, we have water that gets formed in the condensation reaction, and then we get our, so basically you have, you've got to add all these up but take away the H2O. So we have C20, already messed that up, C12, H24 minus 2 is 22, O minus 1, so 6 and 6, 12, 11. So that is our formula for a disaccharide, which not all disaccharides, a disaccharide of 2 hexose sugars, that will be it. So basically just add them up and take away water. Fun. Okay, now we're going on to polysaccharides. This is the major part of it. So we have starch. Here is the the thing about starch. Starch occurs in 2 forms. Or not 2 forms, there's like two constituents is the fancy word. 2 constituents of starch. We have maltose. No. You can tell I'm getting tired at this point by the amount of mistakes. We have amylose and amylopectin. I will draw them. You don't need to learn this, but I just learned it out. Mustava says cellulose is beta glucose. Correct, and we're coming onto that very soon. So amylose, I'm just going to draw out the very basic like diagrams. This is how we're gonna go. So here is our nice wee amylose. And so on and so forth. It goes on like that for like a large number of things. So like repeats to n. So yeah, I forgot about these two bits here. So yeah, you may be asked to label this bond. This is a alpha because amylose is made of alpha glucose. Alpha 1,4 glycosidic bond. That's the name of the bond. And then the cool thing about amylose is the chains come around and kind of just coil up like this. Hydrogen bonding between, like, they hydrogen bond every 3.6, I think. Don't quote me on that. But it's like they hydrogen bond like that, so that it all gets held in place. I think I read that they hydrogen bond every like 3.6, basically every fourth they hydrogen bond together. So it's amylose, then amylopectin. We have it starts off very similar. I'll draw it even smaller. I forgot. Oh. So yes, here we have pretty much the exact same thing. So yeah, they're pretty much the same thing. But remember I said cannot forget about these. Now what I'm going to do is I'm going to go. And now we have we have a chain going off there called a branch. You may be asked to label this. This is a alpha because they're still alpha glucoses. Alpha 1,6 glycosidic bond. Because it is between, so here's the oxygen, so 1, 2, 3, 4, 5, 6, between the 6th carbon and the rest of it, called an alpha 1,6 glycosidic bond. So as we can see, it is branched. Makes it more compact. So together, amylose and amylopectin make up glucose. They form starch. Yeah, I may have said glucose. Starch. They form starch. Starch is found in grains. Isn't that right? Yes, starch. Grains. That is how you must say it. It must be starch grains to get the marks. For like how is it stored or what forms it in. Starch grains is the only acceptable answer. So it's very compact because it's coiled, so you can fit a lot of it into a small space. You can have like quite big starch molecules or starch molecules. They can get quite big in the cell, used for storage. They are insoluble. That is very important. So let me go back to here. We'll do this in red. Okay. Compact because of the coiled structure, helps you fit a lot of starch in a small space. Can get big. Pretty much get big. You store a lot in. They are insoluble, which means, now, are you ready for this? Insoluble means it does not upset the osmotic influx or the osmotic balance of the cell. Basically, you have your wee plant cell here, okay? Here's the very, very basic plant cell. We're not next topic with cells otherwise, but we have starch. I'll draw like some starch. Well, technically starch is found in inside chloroplasts, but so the red dots I've drawn are chloroplasts. So we've got wee bits of starch in here. But because they're insoluble, they will not be carried outside the cell by osmosis, which you shall remember from 5th year. But not be carried out. They will stay inside the cell. They won't make the cell, so they will not. They do not raise water potential. If they were to raise water potential, then they do not lower water potential. That's what I should be saying. They do not lower water potential. If they lowered it, then water would go into to raise it again. But they do not do that, which is good because it means the plant kind of stays as it is. Look at that diagram. Isn't that very nice? There's a wee thing of starch. I'll leave that there for a bit so that YouTube might think maybe I should make that a thumbnail. Okay, let's rub that out and let's move on to glycogen. Now, I have literally just rubbed out the diagram for glycogen. Why? Glycogen is pretty much the same as amylopectin. The diagram is identical. Maybe you want to say glycogen, go for it. Glycogen. So, what does it look like? If you haven't yet, are classic shapes. You do kind of get decent at drawing these hexagonal shapes. Don't have to be accurate, just so yes, cannot forget about the branches, and just like amylopectin, glycogen is branched like that. But the thing to remember about glycogen is more branch. That's not how you spell more. More branched. More branched than amylopectin. So here is the phrase that you rhyme off in the exam. Why? So a question could be like, why is it a good energy source? And I should clarify, emergency, because glycogen is not the main energy storage in animals. So starch is the main energy storage in plants. Glycogen is not in animals. It is lipids or triglycerides that are the main ones in animals. Why is it good energy source? So you want to say that the many branches contain many terminal ends for enzyme activity. So it has many terminal ends for enzyme activity to break it down to supply emergency glucose. So yes, glycogen, many branched ends. It is branched approximately, do that here, every 8 to 10 glucose units. That is how many branches you get. Glycogen is found in glycogen granules in muscles, for example, because the muscles like to, use a lot of it to respire. Muscles also have a lot of, muscles have many mitochondria. Yes, muscles have many mitochondria because obviously you're using muscles quite a lot. But yes, glycogen granules, so starch grains, glycogen granules. That is what you want to remember with that. And that's pretty much glycogen. Now our last one is cellulose. Why is there a wee thing there I can't draw? Cellulose. Okay. So the thing about cellulose, it is not alpha glucose. We have, so if I do kind of like how the textbook draws it like that. So we have OH here, H here, H, OH. And then we have, this is like, whoa. We now have OH, should be an oxygen, H, OH, HO, and so in the condensation reaction we get a water away here, water away here. This is no longer alpha glucose, we now have beta glucose. And what's more, so we have like, I'll draw this diagram, beta, just like next one. That was awful. With beta, I'll go for this one. Beta inverted, beta inverted, basically due to how they have their things, they like to get inverted, which means they can do their thing. So these are just long, very, very long, straight molecules. They're kind of like, just like this. And what they do is they form hydrogen bonds to each other in this wee bit here. And then, well, sorry, this one does not have it there, this one has it here. So they'll form hydrogen bonds, which brings them together into microfibrils. And the thing about the microfibrils is, or the whole thing about this is high tensile strength. And so the role of cellulose is for structure and support. I would always say both to ensure you get the marks, hint, hint. Some mark schemes, I think most mark schemes accept support, but not all accept structure, but say both to get the marks in, and they're found in plant cell walls. And like, what's the difference between cellulose and the other two? Well, the difference is, I am so tired. The difference is they contain alpha glucose, or the other ones contain alpha and this one contains beta glucose. Okay, we have finished carbohydrates. It took us a while, but we got there. So that is the first bit finished. Whew. Yeah. It's like break time for me. Whew. I need a drink. Anyway, our next one is lipids. Actually, lipids are really easy. There is not much to it. So let's fly through lipids. I'm going to need purple for this. And let's put the black back. I should probably be using black for lipids, but you know what, it's fine. So, lipids. Okay, I actually really like lipids, they're nice and easy. Lipids. We have, like, we have a glycerol and 3 fatty acids. Job done. And that is literally pretty much it. Obviously there's more to it. So a glycerol is like this. Those ones are just H's there. And then a fatty acid is like this. And obviously these are all connected hydrogens the whole way along. So we have our glycerol, and this is carboxyl group. And this here is a methyl group. You need to know that, I'll just do COOH, is hydrophilic, and CH3, which is that bit here, is hydrophobic, because there's no charge, it's not polar. So whenever then the fatty acid reacts, what's the reaction? The condensation reaction. You then have, so I'll just represent glycerol like this. Then we have like 3 wee bits, and then we have our fatty acids. What is the bond called? The bond is called, girl's name, Ester Bond. I think that comes from some chemistry thing we haven't learned yet. That's called an ester bond, formed in condensation reaction between a fatty acid and a glycerol. Okay, so then what we need to know, so yeah, and they can be split apart by hydrolysis as per usual. Fatty acids can be very different from each other. They differ depending on how long the chains are, and also whether they have any bends or double bonds. So say we have a nice, it's called hydrocarbon tail, really. Oh look, we happen to have a double bond now. This is like a section of it. There's like loads more on each side. That just keeps going, really. As you may notice, these carbons here can only like have one thing, and because of Vesper theory, basically chemistry says that these will repel each other. So what happens is it like bends. There's a bit of a bend that like pull apart, or all this stuff is coming in here, and then it starts to bend. That means that it's there whenever, like, so you got your wee triglycerides like this. So they're all looking like this, and they can pack nicely into each other, you know. But whenever you've got bends, they can become like all tangled and well, not tangled, but like they just don't stack as nicely, don't go nicely, so therefore they're liquids at room temperature. Oil are saturated or unsaturated. Unsaturated means contains double bond, saturated means does not. The way I remember this is it is saturated with carbs, or saturated with hydrogen, so that is saturated with hydrogen. This is not, because we're missing space here due to the presence of the double bond. No H in that wee thing there. So yes, that means it is saturated. We're unsaturated. Okay. Then we have another type of thing now. If your teacher likes their colours, you might already know what this is. Or if you know the topic well enough, you'll know what this is. So this is a phospholipid. It contains a phosphate group, which is hydrophilic. I probably spelled that wrong, but it's fine. Hydrophilic. And then these three tails, as we know, contain methyl groups, and so are hydrophobic. Okay. Hydrophobic, hydrophilic. They exist in cell membranes in aqueous solution. They don't like water, so that's a bit of a problem, isn't it? So we can also redraw this diagram as this, by the way. Sorry, this should not be here. I'm stupid. Basically, the difference is it replaces a fatty acid with phosphate group. So we can draw it like this. This head here is phosphate and glycerol, and those are the two tails. So what they like to do is I'm just going to draw some equal signs here, and then fill in the heads here. And we have what we call phospholipid bilayer. You'll learn this if you haven't already in the cells topic. That's what cells are made of, nice and fluid. Sorry. So yes. And then also, like, we have our wee hydrophilic heads right here. All the hydrophobic tails go into the center, so they're not in contact with the water. So it kind of looks like this. Then we have another hydrophobic. It's cholesterol. What is cholesterol? It's a hormone, isn't it? But it's like I can't even remember what it is. I knew it at one point. It binds it together, but it's also hydrophobic. Therefore, it likes to be in the middle. And there, it makes it harder. I'll know it whenever we do. I actually revise the cells topic. Lipids, complete. Yeah, if you want. Well, I'm gonna continue. Okay, proteins. I know very well. I actually do know very well myself. So I'm gonna go straight to nucleic acids, and if I have time, I'll come back and do proteins at the end. I want to sleep. Okay, nucleic acids. Woohoo! This topic is actually not too long. Well, nucleic acids isn't too long. So here we go. 3 components of a nucleotide: phosphate, sugar, base. Lewis, hello.
What are you doing?
I'm doing a live stream.
Oh. Is this about time to come in?
Yes.
Is this live?
Yes.
Who's on the live stream?
There are 3 people currently.
Who?
I don't know. Goodbye. Okay. So, 3 parts: phosphate, sugar, and base. We have to use the proper terms now. Phosphate group. It is a pentose sugar and a nitrogenous organic base. And you may know from GCSE that there are 4 of these. They are adenine, thymine, cytosine, and guanine. Sorry for ruining your day, but we also have uracil, and that's the only extra one. You also probably know about ATCG. The A will bond to T, C will bond to G. A and U don't bond, but they are related. And they only ever bond in one situation, which we'll cover. But I'll tell you why AT and CG bond. So, nucleotides come together in long polynucleotides to form DNA. So I'm going to draw them all like this. No, and you may be thinking, well, okay, so yeah, they also do combine in condensation reactions. What's new? But what is the bond called? This time it is a phosphodiester bond. It is a phosphodiester bond. Phospho because there's a phosphate. You know what I mean? Phosphodiester bond. That is what they are called. So yes, they all come together as a nucleotide. These are all bases, they can be whatever they want. And then they will hydrogen bond in the full molecule to other bases. I'll tell you the rules of bonding in a bit. And then what we have, so then we have our sugars. To be honest, you can just kind of draw these like houses. You do not have to draw perfect pentagons in the slightest. Okay, so then we have, I drew these far too far away. Also, I drew them wrong. Classic me. You can tell I'm pretty tired at this point. There goes better. So as you see, one is going this way and one is going this way. This is called anti-parallel. What that means is that they are parallel to each other, but one is flipped. Anti-parallel. And you can kind of see that the space between here, we'll call it the distance, is the same. That's called spatial symmetry. Whenever the bases hydrogen bond, they do so in a way that preserves spatial symmetry. So we have our phosphate sugar backbone, and the only thing that changes in the whole molecule are the bases. So we know that adenine A can only bond with T, and C can only bond with G. But why? We have things called purines, and we have things called pyrimidines. And how do you categorize them? A, T, C, G. That's how I remember it. A, T, C, G, the first one, the last one. Purine. I'm going to draw wee circles. Purines. Pyrimidines. You also can put U in here, the elusive U, which we don't know what it's for yet, but yes. So they look like this. So as you see, there's like a 2 and a 1. The rule is a purine must always bond to a pyrimidine. So look, we have this to a this, and we have this to this. D. And you may also notice that I drew 3 hydrogen bonds in here and 2 in here. So that can actually tell us exactly what they are. This is G and this is C. This is A and this is T. Reason being, G and C have 3 like hydrogen bonding points, 3 hydrogen bonding points, and then A and T have 2 hydrogen bonding points. James McKee wasn't letting them start, thought that we were finishing. I wanted to get it done at home, just like that one question. There's loads more questions in the booklet, but I just did that one at home because I was in the middle of in class and just wanted to get that question done. But yes, we're finishing the booklet in class. She said there wasn't any homework, but we were doing that question in class, so I thought I'd just get those 3 parts finished. Okay, so yeah, James, if you like skip, you'll hear this eventually. How long have I been going for? Like an hour? Let me see, what is it? I don't even know where I find this out. Wie lange ist es? An hour and 9 minutes. Wow. Okay, let's continue. So yes, they must bond in a way that preserves spatial symmetry. The Ds are the same, and also they have different numbers of hydrogen bonding points. That's basically just how they are like made. They'll have different numbers of hydrogen bonding points, which then in turn means that they can only bond with each other in that specific way. So, what is this elusive U? Why is it here? Why do we have to know what uracil is? And get it away from me. Well, luckily for you, uracil is in the less common RNA. Well, actually, RNA is probably more common. Well, hi Jessica. RNA and DNA are... So, let's get into it. RNA, DNA, similarities and differences. So, we are single-stranded here. And we are double-stranded here. So we have like this, and then we have our double helix, in case, you know, most people know that what that's called, but double helix. RNA contains uracil. DNA contains thymine. DNA contains thymine and not uracil. RNA contains uracil and no, whatchamacallit. Yeah. And then RNA contains ribose. This contains deoxyribose. And that is the difference between them. Also, RNA tends to be a lot shorter than DNA. RNA, let's keep on that topic. RNA has 3 forms. Not 6, as that might suggest if you're mathematical enough to understand that 3 factorial 6. Fun fact, it does. Oh, hello. There are 3 forms. We have mRNA, which is called messenger. We have tRNA, which is called transfer. And we have rRNA, ribosomal. Ribosomal DNA, or ribosomal RNA, is just the stuff that makes up the ribosome. tRNA looks like this. They kind of come and here's our RNA chain, or like strand of RNA. They will come and hydrogen bond and then release this. It's kind of like this. They like to release these are amino acids here. They release those and they go off as a wee peptide bond between them. So protein synthesis is actually, once you learn it, it's quite nice. So the DNA opens up. So let's draw the DNA. Oh, time out. What? Father, have we been going for too long? No, apparently not. Are we still live? Are we still working? My phone was like, time out. I don't know if that's a good thing or bad thing. We seem to be okay. So here we have our DNA. What it does is it starts to open up like this. We have all of our bases which are now exposed. And what happens is our mRNA comes and, well, it's like kind of like protein synthesis where it's kind of like whatchamacallit, DNA replication, where it'll come up like this, but then what this will do is this will move off like this. It'll go through the cell membrane. So by cell membranes, phospholipid bilayer, like this, kind of like that the whole way through. So it'll go through there. DNA cannot, it's charged particle. But that'll go through there. and then it'll go to the ribosome, which in itself is made out of RNA, and it like feeds its way in and comes out the other side. It's got all three bases here, like this. And then what happens is, that's the wrong colour, we have our tRNA, which looks like that kind of thing. It has like, oh no, sorry, it only has three bases. This is called the triplet code. It'll come and form hydrogen bonds with one of them and release the thing, and this happens the whole way along the whole way along the mRNA, and you get a protein at the end. That is how that works. And then DNA replication is the final thing, you know, in this, and then we'll go into proteins to finish up. Okay, so DNA. Here we have a blue strand of DNA. Okay, here's a nice blue strand of DNA. I said that too many times. Sorry, that's wrong. I need to do the thing I'm going to do before doing that. Okay, so here is our DNA. It is opened up. Then we get this, and it goes in. This is more DNA, so the bases are exposed. We get DNA from the free nucleotide pool, so there's like nucleotides that are kind of just there. They all join up like this. And so, as you can see, the DNA, like this, kind of happens the whole way along the molecule. Like this, the DNA becomes, so this turns into this. This is page is really not gonna let me draw it. Like this. So now we have DNA. As you can see, it is 50% blue, and it is 50% red, 50% original, and 50% new. And that's how DNA replication works. Shall I just kind of give the rant? So we have DNA helicase, which breaks apart the helix. I've started to get a rash. Breaks apart the helix and unwinds it, breaks the hydrogen bond. It kind of like opens up like a zip, or it unzips. Then there are bases left exposed. The mRNA, or not the mRNA, but it's like mRNA, just the free nucleotides come up like this. They get all snuggly with these two things. We have original, we have new, we have original, we have new, like this. So the 1 becomes 2, like this. Then if we were to repeat the process and we're going to add purple DNA, these bases are going to be purple for diagram purposes. So whenever these two split, we're going to get our same blue strand, Remember, these are all separating, splitting. We also have this red strand here and this red strand here. And each one of these, the purple is going to come up here and here and here and here. So then we get these purple strands like this. So as you can see, each set is, or the whole set in general is 50% of the previous set. So this is 50% of this one, but it's also 50% of the new one. And it is now 25% of the original, and it is 25% of this one, and then also 50% of the brand new one. So that's how that works. People have worked it out by using the Mendelssohn style thing. Just learn it. I cannot be bothered to go over it. It's so boring. It's just like, it's like the process of penicillin. Is that what it was last year? And the whole how penicillin was discovered. So boring. Didn't learn. Okay, proteins. There's quite a bit in proteins. They're easy enough, but I'll do it anyway. So this is a complete video. Proteins. Time out. What is this? Why? Waarom machst du das denn? Are you gonna work? I'd very much like you to work. That'd be very nice. It'd be very kind. That'd be really nice. Really nice. Okay. Proteins. Still saying time out. Are we still live? Apparently so. Okay. Here we go. Let's get through this as fast as possible. Proteins are long chains of amino acids. Amino acid looks like this. Usually I do this in colour. There's an amino acid in normal body pH. This happens. When did I do this? This is so familiar. Oh, amphoteric. That's what it was. So yes, the amino acids look like this. I'll undo this and then have it like this. Okay, so here we have an amino acid. As you can see, there is some kind of strange R group. Cadera. What is the R group? It can be, well, there is at least 20 different things. It could be an H, and you get glycine. It can be CH3, and you get alanine. It can be CH2SH, which would give you cysteine, etc., etc. But it's the R group that differentiates the kind of amino acid. Whenever two come together, I will draw the end of another molecule. Here we have our HNCRHC, and like this. As you can see here, we happen to have an H2O. What? Condensation reaction releases the H2O and makes a peptide bond. This bond here, which is called a peptide bond. Peptide bond. Amino acids are long chains of, oh, it's called polypeptide. Dipeptide is two. Many of them is polypeptide. And releases water. You can break it apart by hydrolysis as per usual. We have many structures of protein. There's 4. Primary structure is literally just, it's called it is the sequence of amino acids in the polypeptide chain. Literally abbreviate to that. So kind of like, so we always start off with the end of it. So we have an NH3 plus bonded to lysine. No, this is just completely random. Glutamic acid. Alanine, cysteine. Have another lysine. What else do we have? I'll go for another alanine then. Leucine, alanine, and then we have a COO minus. We have our N terminus and our COO terminus. Terminus means like end. So we have our 2 ending points. That is literally the sequence of amino acids in the polypeptide chain. That is primary structure, no more, no less. So then we have our secondary structure, our secondary, it is called secondary structure, well done me. Secondary structure, there are 2 forms, or secondary structure is basically interactions between primary structure in the form of hydrogen bonds. We have two different kinds. We have alpha helix, where the hydrogen bond, kind of similarly to, what do you call it from earlier? The amylose hydrogen bonds. And then we also have beta pleated sheets. The hydrogen bond kind of like this. Alpha helices are a lot more common than beta pleated sheets. And beta pleated sheets have been linked to diseases caused by prions. But those are the two kinds of secondary structure. Now we go into tertiary structure, or like third level. Tertiary structure is how secondary structure interacts due to our groups. So the protein kind of forms like this kind of thing. We have many different bonds which hold it in place. For example, we could have like Na for some reason, and then we could have Cl. We don't, because that doesn't exist. There is no Na and there's no Cl in the R groups. But you can have ionic bonds. There are some ionic bonds that form. You can have disulfide bridges, so we've got S and an S. Disulfide bridge, disulfide bond. There are also some hydrogen bonds between them, the groups. We've got these things called hydrophobic interactions, which is where, because they don't like water, it's kind of like we have like all these big things. That's kind of hydrophobic interaction. Oh, that doesn't look good. Huh. That did not look good. I got something that's like massive error. Are we surviving? Are we still alive? Seems fine. We may cut out in like 5 seconds. No, we seem fine. Okay, so those are the different bonds that can occur in the tertiary structure. And that all holds it together in a 3D shape. Are there any other bonds I forgot about? I don't think so. I think that's it. So yes, further folding of the secondary structure. Then we have quaternary structure. So we have quaternary structure. This is aggregation of 2 or more polypeptide chains to form one functional macromolecule. So, for example, we have what is this? Well, maybe it would help if I drew in these. Who knows what this molecule is? It's hemoglobin. It contains 4 chains. We have 2 alpha chains, 2 beta chains. They're called. They contain prosthetic groups. Those will be these things made of which contain iron. Prosthetic groups. A prosthetic group is a non-protein, not a non-protein thing that enables the, what's this called? Protein to carry out its function. Helps to carry out its function. It's called a prosthetic group or a conjugated protein. So we have two kinds of proteins based on structures. We have fibrous proteins and globular proteins. Fibrous proteins we have things like collagen. It is, see if I could, there's also, you can also draw it like this, which it's hard to draw it like. And then like this third chain emerges from back, kind of like that. That's an awful drawing. But collagen, it's a triple helix. It only contains secondary structure. Does not contain anything but in secondary structure. And it has great tensile strength used in bone, muscle, tendon, things like that to hold it together. Very strong, does not stretch when tension is applied. Then we have globular proteins, like hemoglobin. They have a metabolic role, in, for example, enzymes, things like that. Hemoglobin is globular. And they do have quaternary structure, and they're often conjugated. Different things can be conjugated to make different kinds of proteins. We can get lipids to make lipoproteins, carbohydrates to make glycoproteins, nucleic acids to make nucleoproteins. Anything you want really can be added. And I kind of want to finish by 8 so I can sleep. Our last thing is prions. I am exhausted. Seriously, this is so tiring. Okay, so prions, represented by P or PSC for the prion form. Prions are basically proteins that are misfolded in secondary structure. They contain increased number of beta plated sheets rather than alpha helices, so they decrease. They're infectious, so if say you have this thing here, this could be a prion because look, it has some beta plated sheets. It comes over and here's a nice wee protein. As soon as that touches that, that will get replaced with beta plated sheet. That will get replaced with beta plated sheet, and it'll become infected to prion. Prions are found mainly in nerve tissue, and whenever everyone has prions, but some of them aren't dangerous. They're like the C one, means cellular. Basically, they could become dangerous at any point, but everyone has them, so nothing really to worry about. Incubation time is 5 to 20 years, so if you have a prion disease, you're not going to know you have it for 5 to 20 years, which is quite a long time, as we all probably know. So yes, prions, they're dangerous. You can get them from like just spontaneously changing, or you can get it from mutations in DNA. So it codes the prion, so it can be inherited, or you can also get it from eating infected beef, usually meats, but usually beef. Found in cows can get it, mad cow disease, bovine spongiform encephalopathy. People can get it, variant Creutzfeldt-Jakob disease, it's the same thing. Scrappy in sheep and goats. But yes, basically what it does is it goes into your brain, and then the prions take the place of like nerve tissue, eat away at them, don't work. You could die. You lose all function and you die. How morbid. I am extremely tired. In case you haven't noticed, thank you for watching the live stream. There are currently 3 people watching. Hi, thank you. I'm actually just going to go to bed because I'm so tired, so thank you all for watching, and I'll see you all next time. Goodbye.